Okay, so of all the parts of aerobic respiration, I love to talk about electron transport chain the most. It's my favorite. And it's the one that I ask the most questions on the exam also, because glycolysis, you've got a big list for that. Krebs cycle, citric acid cycle. I don't focus too much on it, but I do put a lot of focus on electron transport chains because these concepts also apply to how photosynthesis works. And they also apply to how anaerobic respiration works. So one thing that I want to mention here is some terminology. So this process of having an electron transport chain and passing electrons at the end of it onto a molecule, we can call that respiration. So if we're referring to metabolic respiration, we're implying that there is an electron transport chain and there is an electron acceptor at the end of that process. So if the final electron acceptor in the process is oxygen, we call that aerobic respiration. That is the actual meaning of the term aerobic respiration, is there is oxygen at the end of an electron transport chain to pick up electrons. And along the way, that made a lot of ATP. If the final electron acceptor is something else, anything except oxygen, not oxygen, and there is some kind of electron transport chain involved, that's anaerobic respiration. Anaerobic respiration. Several people put on your reading quiz that fermentation was anaerobic respiration, and it's not. It's anaerobic, but if we're saying respiration, we're talking about an electron transport chain operating. We cannot do this as people. We don't have any other final electron acceptor besides oxygen. Bacteria can, but if you refer to anaerobic respiration, that's not the same thing as fermentation. Just wanted to get that out there. So when we're talking about the respiratory machinery, we're going to be looking at certain proteins that are found in the inner mitochondrial membrane. So this gray box is the inner mitochondrial membrane, if we're talking about where people do this. And these proteins can do three cool things at once. They can receive electrons from a donor. Some molecule is going to give them electrons. And when they get the electrons, they become reduced. That's a thing that you've heard of before. And when they get the electrons, they are then going to pass them on. So they're going to act as electron acceptors and electron donors, or oxidants and reductants. Boom, boom, back to back. And when they pass the electron on, there has been an energy drop in the electrons itself. So I think of this like hot potato. This is a silly old game. Like, you get something, oh, it's hot, I pass it on. By the time I pass it on, it's cooled off a little bit. It's lost a little bit of energy. Proteins that are involved in electron transport chain can take that energy and use it to pump protons across the membrane. That's the cool part. That's what makes them the most special. So yeah, I can receive electrons, I can pass on electrons, and I can pump protons from one side of a membrane to another. I can help form a proton gradient. See if you remember, where are the protons accumulating in the mitochondria? Is it the matrix? It's the intermembrane space. That's going way back, or something that you haven't read about lately. But this is an example of the protein I was just talking about, something that's involved in electron transport chain. It's picking up electrons from the matrix, because that's where they are right now. That's where NADH and FADH2 are. They're in the matrix. They're dropping off electrons on the matrix side, and those proteins are using that energy to pump protons into the intermembrane space. So we get out here a proton gradient, and it also happens to be very acidic out here. We've got a proton gradient, and they are stuck. Protons can't go through the membrane. They're charged. They're stuck out here. The only way that they can come back in is through an ATP synthase protein. And they do come back through, and ATP synthase uses that rush of motion to produce ATP. So this is a picture of how oxidative phosphorylation works. So it requires a gradient. The gradient of protons is formed using electron energy. The cool part is having these electron transport chain proteins move electrons and pump protons at the same time to make that gradient. So that's a little introduction. This is happening in mitochondria in higher organisms. Lower organisms can do something like this, but they use their cytoplasmic membrane to do it. So prokaryotes have systems like this. They're just doing them in the cytoplasmic membrane, and sometimes they're not using oxygen at the end of the line. But we do this in mitochondria. And the more energy a tissue needs, the more mitochondria it will have. So muscle tissue has more mitochondria than other tissues because it uses so much ATP. It needs more ATP production capacity. Sperm tails are actually wrapped in mitochondria, which is kind of weird, but kind of makes sense. They need so much ATP to spin the tail. They have mitochondria wrapped all the way around. So to review the compartments, here's a transmission EM of a real mitochondrion. And it's got an outer membrane that you could trace around, and it's got an inner membrane. And those two membranes are dividing two different compartments. This is a little bit hard to look at, so I need to go to a different picture. Let's use this picture. So here's the outer membrane. What's out here? Cytosol. Cytosol is out here. Glycolysis is out here. Here's the outer membrane. Here's the inner membrane. The inner membrane has a lot more surface area than the outer. The inner membrane is folded up a lot upon itself inside the outer membrane. So if we go back to the real picture here, we've got outer around the outside, and the inner is really contorted. So this, if I follow this around, here's the inner, comes all the way down in here, goes back, out here, goes back, goes back. It's got a lot of surface area, and that's a good thing for the mitochondrion because the more surface area, the more room it's got to hold electron transport chain machinery. The more membrane it's got to pump protons across. More surface area for producing energy. So the space in between that is the intermembrane space. Between the membranes, intermembrane space, and that's where protons are going to be accumulating. Inside the inner membrane, that's the matrix, and that's where citric acid cycle was going on. That's where we have a lot of NADH. There's a lot of acetyl-CoA pyruvate in there. That's in the matrix. It's important to note here how these membranes are a little bit different from each other. The outer membrane is a little bit more permeable than your average membrane. So anything that's smaller than 5 kilodaltons, anything that's smaller than 5 kilodaltons can go through that membrane. It's fairly permeable. The inner membrane is kind of more like any regular membrane. So it doesn't have largish holes that will let things through. It better not, because it needs to be able to exclude protons from crossing back over anywhere except ATP synthase. It's a good thing for the cell that the outer membrane is kind of permeable, because there is so much important metabolism going on in here that trying to get everything to cross through 2 membranes is too energetically expensive. Anytime you have to move something across a membrane, it has a cost to the cell, and we're going to see why that matters on Tuesday. So one of the things that you have to memorize for your test next week, and the reason I wanted to go ahead through this today, is I want you to memorize the names and the order of the components of our electron transport chain in people. And there are 5 components. So we have the big green blobs. Those are actually the ones that pump protons. They expand both membranes. There are 3 of those. The first one's called NADH dehydrogenase. NADH dehydrogenase. There's actually another one that's not shown here. We'll come back to that in a minute. This is also called complex 1. Some people will call it complex 1. If you work in Dr. Fitz's lab, they refer to these by number. So this is also complex 1. There's a complex 2 that's not shown on here, and that's my biggest heartbreak with this book, which I love for every other reason. There's a complex 2 that's shown on here. And that one, I think it's called, I'll mention that to you next week. I want to get the name wrong. The third component is not a proton pumper, it's a small molecule. It's called ubiquinone. Ubiquinone. Then we have another proton pumper. This one is cytochrome BC1 complex. It's also complex 3. Can have either name. I use these names. I use the book names in here. The next in the chain is cytochrome C. So that's useful. We have cytochrome BC1 complex on one side of cytochrome C, cytochrome oxidase on the other side. And that makes sense because this one is going to be taking electrons off cytochrome and oxidizing it. So we have cytochrome oxidase. This is complex 4 as well.
There's 2 complex 4s.
Let's see. 1, 2. This is 3. This is 4. Did I say that this one was 4? This is 3. This is 3. So you need to memorize their names and their order. This one is first, and its name tells you what it does. NADH dehydrogenase. This is the drop-off point for electrons from NADH. It's also complex 1. Complex 2 is in essence the drop-off point for FADH2 electrons. Ubiquinone is a small molecule, not a protein. Cytochrome BC1 complex receives electrons from ubiquinone, donates to cytochrome C. Cytochrome oxidase picks up electrons from cytochrome C, and this one is going to be passing electrons on to oxygen also. This is the end of the line for the electrons. And that's our electron transport chain. I do not consider ATP synthase to be part of the electron transport chain because electrons don't pass through it. But I think in some places it's called complex 5. It is not picking up electrons. I think that's confusing terminology, but it's here. It's involved with the process of making ATP. It is not an electron acceptor. And I think that's why your book doesn't put it on here. So that's your memorization task. And the last slide I'm going to talk about today is here. So it's referring to what I just said. So here's electron transport chains. Electrons are arriving at one end, being passed down the line, eventually being added onto oxygen. This is aerobic respiration. So ubiquinone is here, cytochrome C is here, there's complex 2, not shown here. Electrons get passed onto water. During that process, protons have been pushed across the membrane. They've accumulated here. They are bouncing off of each other. They're very crowded. They can flow through ATP synthase. That's where the ATP gets made. But ATP synthase is not getting any electrons. Doesn't have anything to do with electrons, but it is letting protons rush back through that were pushed across the inner membrane using energy from the electrons. It's involved. It is not touching the electrons. The electrons are the red line. So that's an intro to what we'll be going through on Tuesday. And on Tuesday, I talk about aspects of how does that actually work. How does that actually work? But I wanted you to go ahead and start on the memory work before Tuesday. Before Tuesday.